you have a little bit of background knowledge on. So you should be able to move along quite okay. Okay, so today we are going to be talking about how cells obtain energy. Um, so specifically, we are going to be talking about how, once we have the food that we consume, what do we do with it to get energy to be released from that? And in that case, what we are usually breaking down is actually the glucose molecules or the sugars that we have in our foods to get energy from them. So plants obviously make their own sugars by photosynthesis from CO2 and the water and the stuff they get from sunlight, the energy they get from sunlight. Animals, on the other hand, are eating this food and then breaking it down. But whether you make your own food or you consume food that is already made, you are going to either way oxidize these sugars and break down the bonds in them to release the energy that is stored within those bonds. And that's what we use for all our processes at the end of the day. And the bonds that we break are usually the ones that are at the end of the molecule and are termed as the high energy chemical bonds. And we'll talk about those in a little bit. So cells in our body obviously need some way to break down these bonds. The process to break down these bonds, although is going to be something that can happen spontaneously as well, will typically require higher temperatures to break them down in the proper optimum manner. So in our body, we have to use enzymes instead to break these sugars down in a series of controlled reactions that are paired up, just like we talked about last week, or last week on Thursday, where they are exogenic and endogenic reactions are paired together so that you can use energy from one to fuel the other. Which one are we going to fuel?
What was the question exactly, Professor?
So you know how we have different types of reactions. We have exothermic and endothermic reactions. Which ones do we fuel in our body in, when we couple reactions to each other? Which one provides the energy and which one is fueled by that energy that is released by the reactions?
It's endothermic that creates the energy, and it's exothermic that excretes the energy, right?
Anyone else? Let me get
the exothermic provides the energy and the endothermic takes it.
Yes, it's the opposite, Taylor. Yes, the exothermic are what provides the energy because they release energy from the in the process of the reaction moving forward. And then that energy is then taken in by the endothermic reactions to make those processes able to move forward. Taylor is in the middle of the sun, getting all the energy from the sun. Unable to work.
Right? My eye.
I know, I saw you completely like closing up your eyes. So yeah, and so you'll see that, throughout this process of breaking down food to release energy that is in them, you're also going to have reactions that require energy input before they can move forward. So you don't have every single step in the reaction as an exothermic reaction where it's releasing energy, but rather you still have coupled reactions that overall are going to provide you with a lot of energy that you can use. So here it is showing you that if we had a direct burning of sugar in a non-living system, so we could do it in a chemistry lab or another setting, you could use oxidize it with the oxygen, and you would need a lot of activation energy input to begin with so that you could release the energy that is stored inside those bonds. And then all that energy is going to be released as heat. Nothing is going to be stored. And the products that you will get at the end of the day would be carbon dioxide and water. In our bodies, you do a series of reactions to get to the same endpoint. So the same input and the same output. You still have your sugar molecule and your oxygen as your input, but now you break it down one bond at a time in a series of reactions, so that each time you release a little bit of the energy, some of which can be stored in activated carriers, whether they be GTP, ATP, NADH, whatever type of molecules are going to be available at that time. And then as a result, you still get the same products. So it's not changing the reactants, it's not changing the products, but by doing it in a stepwise manner, one, you can work at body temperature, because each one of those mini steps don't require as much of this activation energy and are able to move forward in the normal physiological temperature. And we use the enzymes to kind of give them that mini environment so they can happen. And each time you are using some of those activated carriers to store some of the energy. Okay, so when we look at food, obviously it's not all going to be sugar. Some of it is going to be fats, some of it's going to be proteins. Each one of these categories are going to be breaking down differently at the end of the day, right? Now, whether you have you're consuming proteins, fats, or polysaccharides, which are just long chains of sugars, either way, in order for them to be part of this breakdown of process that leads to the ATP, they still have to go into the same system, the same set of network. So it first, if they aren't sugars, they first get broken down into their smaller monomers, which are the individual amino acids or the fatty acids and glycerols. And then they are then converted into one of the intermediate steps. So they can then be used in the same citric acid cycle or in the same general catabolic cycle related to sugar breakdown, releasing the amount of ATP that we need. Amino acids get broken down into, depending upon what their side chains are and what type of subunits are composed of that, they will get broken down into pyruvate and acetyl-CoA, that they can then continue the process just like normal sugar would. Fats get also broken down into acetyl-CoA before they can get shuttled into the same pathway, where they can then go through the citric acid cycle and the electron transport chain as well and release a lot of energy. However, sugars first have to be broken down through a process called glycolysis to be converted into pyruvate molecules, and then they work through the same system. So we're going to go through the various processes that are needed to break each one of these categories down so that we can release energy from these systems. So there are 3 stages of catabolism, which is basically degradation of these macromolecules into their monomers, and then eventually to release energy from the individual bonds that are present there. The first one is digestion. Obviously, that's at the big macro level. And that's when enzymes within our intestinal tract and within our stomach are digesting and breaking the food down into their individual monomers and into smaller subunits that can easily be utilized by our body. So at that point, what you will end up with are your fatty acids and glycerols, your simple sugars, and the amino acids. And they can then be further catabolized through our system. Now, in glycolysis, you are going to use all those simple sugars, all the glucose that you have gotten out of your food, and you're going to break that down further into 2 smaller sugar molecules. In the case of pyruvates, which are actually just 3 carbon molecules instead of the 6 carbon sugar in the presence of glucose. And then your amino acids and fatty acids will go through their own breakdown system to get to acetyl coenzyme or pyruvate system. They then get shuttled into a citric acid cycle, which is where a lot of these activated carriers come in to take some of the energy from the bonds as they're getting broken down. So in citric acid cycle, you are going to have the pyruvate and the acetyl-CoA molecules getting broken down and then remade in order to provide reduced electron, reduced electron carriers with extra energy from these electrons. And then those carriers will then get shuttled into the mitochondrial inside matrix and go through the electron transport change, which is where majority of our energy is produced through oxidative phosphorylation. So you should know basically the catabolism is the process through which our body releases energy from the food that we are taking in. It has the three processes, digestion, glycolysis, and then finally the citric acid cycle and electron transport chain, which is where majority of our energy is made, is right here in this electron transport chain and the citric acid cycle. You should know that majority of the those activated carriers are charged essentially with this extra energy from the electrons, broken bonds in the citric acid cycle. And all those charged carriers are then taken into the electron transport chain where they're used to create ADP. Cool. Okay. So the first part that we're going to look at is splitting of a simple sugar in the form of glucose into the two pyruvate molecules and how that process happens. Now, this particular process, the simple glycolysis, occurs whether or not there is oxygen around in the system. And it does produce some small amount of ATP as an output as well. So the word glycolysis simply means, glyco is with the sugars and lysis is splitting or breaking apart. So it is a very, it literally means exactly what it does, sugar splitting or sweet splitting. It occurs in the cytoplasm, so there's no mitochondria involved in this first step. So as our cells take in the glucose from our body, from that passage from the intestinal lumen into the cells, they are able to, or from the blood into the cells, they're able to start to digest it and break it down further into these pyruvate molecules without having any need for extra oxygen or anything else. However, this does require some energy input to start that process along. So this one is going to be requiring some energy input from us. So you do need to have some ATP available already in the system, right? So this is what you would call an endothermic process. It does initially to start that process. However, once you split the molecule into the two pyruvates, which are these two carbon chains, you end up not only getting back those ATPs, but also two additional ATPs and two NADH as a result. What's NADH? So yes, it is another form of an electron carrier. So this gets activated. This is now two activated carrier molecules that also contain energy in addition to the two ATPs that you've gotten as a result. So even in this simple process, just at the very, very beginning, you're already getting some minor amount of ATP produced or energy getting produced that can then be used to fuel further processes, whether it is to break down more sugar molecules or to do something else in our metabolic pathways. Now, within this process of glycolysis, there are several steps involved before you get these pyruvate molecules. In total, there are actually 10 steps involved. So these are 10 individual steps before you can release any type of energy from them. So your initial glucose molecule first takes in individual ATPs, which are your individual steps. And each time they're using them to create phosphorylation on one of the carbon chains, so that in the end you have a fructose 1,6 bisphosphate molecule instead of the glucose. Now these charged so-called carbon atoms at the end at this point, because of these additional phosphorylation, allow it to get broken down. That creates that high energy bond over here that can then be broken down in the next two steps to individual chains of 3 carbon sugars with a charged phosphate group on one of their ends, which then is cleaved at the next few steps to release your charged NADH molecule and your 2 ATPs as a result. Now, each one of these 3 carbon chains will give you 2 of the ATPs and an NADH molecule. So step 6 through 10 is where you're generating the energy. The first 3 steps are where you're investing the energy by taking 1 ATP at a time onto your initial glucose molecule to charge it so that it has some unstable kind of bonds that can then be cleaved. Those bonds are then cleaved to create your 3 carbon sugars or 3 carbon chains. And then those 3 carbon chains are then used to generate energy by breaking down their high energy bonds in this case. So at the end of the day, you have removed the phosphate and you have changed the bonds, the end bonds with the oxygen and the hydroxyl so that you can have 2 pyruvate molecules at the end. You do not need to remember all 10 steps. What you do need to know is that step 1 to 3 is your energy investment phase. Step 4 through 6 is where you are breaking down that initial, you should know this one. You should know that you create a fructose 1,6 bisphosphate at the end of the energy generation. And you should know that the energy is then released with because each one of these initial 3 carbon chains have that phosphate group on them that gives it that high energy bond that can be broken down easily. And so then you should know that the next 5 steps are where the energy is generated in the form of 2 ATPs and an NADH per chain, giving you your final net output of 2 ATP and 2 NADH for every single glucose you put in. Cool. So you should know the basic reaction and just which parts are energy investment, which parts are charged molecules, and where you're generating your energy and how much. Sorry. Okay, so moving forward. So again, just like before, these processes would not go on unless they were coupled, since some of these are not are going to be endothermic and some of these are going to be exothermic. So if you looked at these reactions up close, you will notice that even though step 6 through 10 are termed as energy generation, they are coupled because some of the steps within them actually are not energy generatable themselves. So you have, if you take the energy from step 6 and 7 together, it is favorable. But if I was to just try to do step 7 by itself, it would not be able to move forward because it did require ATP input in order to break this hydroxyl bond at the end once the phosphate was removed. So we couple them by taking that high energy phosphate bond, breaking that down to release the energy that can then be input in the next step to break down the next bond. Okay. So the hydrolysis, I need to move. Sorry, my webcam is always like in a weird place, so I can never tell which slide I'm on. I will have to figure that part out later. So hydrolysis of just the ATP always provides a little bit of energy that can then be used into the next reaction that can then be fueled with it. It is also, ATP is in general very efficient at donating the phosphate group because that third phosphate is its highest energy bond. and it is able to cleave off very easily, releasing a lot of this energy that can then be used to fuel reactions. So this is just showing you the delta G prime for hydrolysis of a phosphate bond. And you can see that anytime there's an ATP, it's releasing a lot of energy. Or anytime there's a phosphate, it's also going to release, attached to another one of these molecules, it's going to release a lot of energy when that bond is broken. The bigger the molecule at the end of the day that has these phosphates, sometimes you get even more energy from them, or majority of the times you get more energy released from them. So if you were looking at just glucose 6-phosphate is only negative 17.5, but when you have those 3 chains, each one with their own phosphate, the 1,3-bisphosphoglycerate, they have 3 times the amount of energy almost getting released from those. So now we have 2 pyruvates. At this point, it depends whether we have oxygen available in the system or not in order to get more energy released from these molecules. If there is no oxygen available, or if there is not enough oxygen available, electron carriers cannot be going through the electron transport change because they won't have any oxygen to use for the oxidization process. So in this case, what they can do is they can still produce some energy in an anaerobic process, which is called fermentation. In this case, they are not going to go through the electron transport chain itself, but it is going to work in a different way to break down the glucose to get some of the energy out. And so we are going to look at how those electron carriers then work in the absence of oxygen in order for glycolysis to continue in the next slide. So in fermentation, what you end up doing is that you take your pyruvates and you use, you again are going to be generating some ATP, but the majority of the system that you're going to use is going to be through NADH. So what you will do is you will take in NAD ions and you're going to convert them into NADH, and then also some protons available according to that. So you just regenerate NADH and NAD plus in this process as you break down the pyruvates into a smaller molecule, into 2 lactate molecules, and then continue with that process through that. So in this case, what you get buildup of in your muscles or in your body is the lactate molecules, which cannot be further broken down without the oxygen. And so that can cause a problem. So obviously it has to be broken down into something other than what you would typically see. And so in this case, you can then use a process to further break this down into an alcohol and carbon dioxide as a result. And so that's the process that you see over here, where this first part is exactly what you saw over there, that you have these lactates that are getting produced. But in addition, what you can do is you can break these pyruvates down, again, using the NAD plus regeneration system to produce some NADH molecules for energy. And also, as a result, break this down into an acetaldehyde, and then break down further into ethanols, releasing carbon dioxide and ethanol molecules as a result in the body. Now, none of this is going to be as high energy output as a electron transport chain. It's going to only give you, 2 ATP molecules and a few of the NADH to work with, compared to a massive release of energy if we were to do appropriate electron transport chain. However, many systems, especially many types of bacteria, do use this method to generate energy for themselves, and especially those living in anaerobic environments. And they, in general, will produce different types of ethanols or alcohols as a result of the breakdown of their pyruvate molecules, depending on the type of bacteria that you're looking at. Cool? Yes, no, maybe? Now, obviously, we as humans would like to generate more energy, and general organisms requiring oxygen can use a lot more or can make a lot more energy. And they can do that because of the presence of mitochondria. So before we talk about how mitochondria is going to help with the system, we're going to recheck and review the structure of mitochondria. So our mitochondria essentially has several membranes and several compartments that are very different from each other based on their biology inside and the membrane receptors inside. Many of the different molecules that we are working with can be organic molecules, can be converted into acetyl-CoA, and then later on can be provided into the electron transport chain to generate a lot of energy. So if you look at the outer mitochondrial membrane, it's going to be a lot more similar in some ways to other membranes, but it is extremely impermeable to outside sources to maintain the environment inside that it needs. It then has a second matrix membrane that kind of convolutes in and out of the inside space to create a lot of surface area within that inner matrix. And in between, there's an in between the two layers is an intermembrane space that specific reactions can happen in. The acetyl-CoA generation typically happens in this intermembrane space and the outer mitochondria, as the molecule pyruvates move from the outer mitochondrial membrane into the inner intermembrane space, they get broken down into the acetyl-CoA and then finally go into the matrix for the last part of the reaction, which is the electron transport chain. So the pyruvates are converted to acetyl-CoA in the next step as they get shuttled into the mitochondria. This involves, again, oxidative decarboxylation, in which one of the carbon is lost from the pyruvate molecule to create an acetyl group. And then the pyruvate is oxidized to become the acetyl-CoA by binding to a coenzyme molecule. In this process, you get some NAD plus that is reduced to NADH and some protons that are released. All of these, again, electron carriers will then get shuttled into the electron transport chain as it moves further in the process. Fatty acids are also used in this process to create acetyl-CoA. In this case, again, they first have to be broken down into their individual monomers. So each one of the, if you have a triacyl glycerol, in the first several steps, you will require ATP to break them down to get individual fatty acid chains that can then be activated to coenzyme A to create the acetyl-CoA molecule at the end of the day. These are, again, using ATP to create charge groups that can then bind to an HS-CoA to form this highly charged group that can be oxidized through the through the citric acid cycle to create coenzyme A that can be used in the next system. So in this case, this is a fatty acyl coenzyme that enters the cycle. And throughout the process, it's going to release FADH and NADH molecules as electron carriers, activated electron carriers, before you get the final acetyl-CoA enzyme removal. Again, in this one, what you need to know is that your triacyl glycerol first has to get broken down into individual chains, and that requires input of ATP. And it gets charged by the addition of an acetyl-CoA group to the end that can then be, and this fatty acyl-CoA enzyme is going to enter the cycle, the citric acid cycle. And as it gets shortened into the fatty acyl to a 2-carbon group, you will release an acetyl coenzyme and you will release FADH and NADH molecules as electron carriers. Okay, you don't need to know exact structures, but you do need to know the individual kind of steps. Again, in the citric acid cycle, you will notice that there are many, many steps again. There are over, again, about 8 steps each time they go through the cycle. And you're going to be releasing 3 NADH and an FADH molecule as a result. You also produce some GTP, which is another nucleoside with triphosphate attached to it. So each time you go through the cycle, you will release 3 NADH, 1 GTP, and 1 FADH. Okay, all these NADH, GTP, and FADH are again activated electron carriers that you're working with. So looking at some of the NADH we talked about before, looking at some of these other. So in GTP, just to make sure that you recognize those structures, it is very similar to an ATP. It is, instead of adenosine, it's guanine triphosphate. So it still looks the same. It's a ribose sugar, guanine, and then the 3 phosphates attached to the 5' carbon on the ribose sugar. It is normally seen as a GDP. When activated, it adds that additional phosphate to create a GTP. In an FADH molecule, you will see that the FADH2 activate once it requires 2 electrons and 2 protons input to create the FADH2, the activated molecule, that is then again taken in by the cells or by the inner matrix of the mitochondria to the electron transport chain. Okay. Okay. So glycolysis had given us 2 pyruvates, right? So when we were still in the cytoplasm, once you get to that pyruvate, it also goes into the mitochondria from glucose breakdown, and it will then again do the same thing. It will go through the same citric acid cycle, and it will produce the same types of the molecules, the 3 NADH, the 1 FADH, and the GTP as a result. However, this citric acid cycle is getting input from everything, right? It's getting input from the fatty acids, it's getting input from amino acids as well. So these are the basic step of glycolysis along with the citric acid cycle is what's going to provide our body with all the building blocks to make other macromolecules like other proteins, because it's breaking down those amino acids and releasing them and then taking the energy from them into the citric acid cycle. It's breaking down the fatty acids, the triacylglycerols into individual fatty acids that are then released into these once they go through the fatty acid cycle or the citric acid cycle, they are released as smaller monomers of the fatty acids, plus the acetyl coenzyme. And they are also providing our body with the energy through breakdown of the glucose molecule as well. So it is the glycolysis and the citric acid cycle that provide our body with the building blocks needed to create more macromolecules for our system, whether it be our membranes or hormones or amino acids in the to create proteins or to create sugars. Either way, all of those are going to be taken in from there. And you can see some of those places where these molecules, these building blocks are created. So in the process of glycolysis, you are going to have these glucose 6-phosphate that can be used to create nucleotides. You are also going to produce fructose 6-phosphate that can be used in amino sugars as those glycosylations that go onto your protein chains to create glycolypids and glycoproteins. You will also produce some lipids in the form of dihydroxyacetone phosphates that can be used in lipid formation to change their groups. You will then, within the cycle, once you get to the citric acid cycle, or once you get to the pyruvate itself is used to create alanine amino acid as part of the alanine amino acid side chain. And 3-phosphoglycerate is used to create as a side chain for serine. So you can find individual molecules all throughout these steps, at each one of these many steps that are used in our body as building blocks to create other proteins or other molecules, other macromolecules. Similarly, within the citric acid cycle, each one of the intermediate steps give rise to building blocks that can make other types of molecules, whether it is cholesterol and fatty acid chains, or again, R groups for other amino acids to be made in the form of purines or chlorophylls or pyrimidines. Cool. So again, you don't need to know exactly what comes where, but you do need to know that different types of molecules are made that can act as building blocks for macromolecules within our body metabolism, metabolic pathways, and you should be able to give examples of a few of them. So if on a question I ask, give me example of 2 different building blocks that can be created through the process of glycolysis, you should be able to come up with, well, it creates sugars that can be added to create glycolipids and glycoproteins, and it produces R groups that can be added to create different amino acids. Got it? Okay. So now we get to the final energy generation, which is where majority of our energy is going to be made, and that is the electron transport chain. And this is which synthesizes the majority of the ATP that is made in our cells and used by our cells. So once you go through once the pyruvate molecule enter the membrane and start to go into the inner mitochondrial space, they first go through the citric acid cycle in that space. And as those molecules are getting, those activated carriers are getting released, they start to go into these proton pumps and a series of carrier molecules and receptors within the inner mitochondrial membrane to in a process called oxidative phosphorylation, where they take their charged molecules and they kind of pass them along like a relay to produce a lot of ATP through these proton pumps. In this case, ADP is coming in as adenosine diphosphate, and phosphates or charged phosphates are added to them to create the ATP that we then utilize for energy. So the catabolic and anabolic reactions are, like I said, they're usually coupled so that and organized so that they can be both regulated and maintained in the level that we need. They are inbuilt control systems that allow the enzymes to regulate and coordinate the rate of our metabolism and the individual metabolic reactions happening in the cells. And they automatically provide a feedback regulation for our biochemical pathways. So when you have a lot of storage of food, it can be broken down, not all at once, but rather in a controlled manner as needed by our body, and not just be utilized all at once so that, we may spontaneously combust instead, which would be kind of scary. If we were to look at glycolysis and citric acid cycle, we notice how it was already over almost 20 different reactions that happened in that process. However, looking at the larger metabolic network in our cells, that's a very small amount, small number, fraction of the reactions that are happening in our cells. There, at any given time, there are many, many, many other processes that are happening, thousands of other reactions that are happening at the same time, all of which require some ATP input, some kind of energy input in the form of those NADH, GTP, ATP, you name it. So this process, while it is a small fraction, is probably one of the ones that is happening the most in our cells to maintain all these other processes, right? That are required for our daily survival and our daily work. Okay, any questions till here before I talk about gluconeogenesis? So while this is a fascinating topic, it is one of those that I find hard to teach sometimes, just because it has so many individual steps and so many different chemical processes involved that are hard to sound interesting individually, right? I can tell you a lot of cool things about protein. What are proteins walking across a myosin or an actin filament? I cannot tell you as many cool things about this, but these are essential processes that we must know in order to understand how our body is able to work the way it does. Okay, so we'll go back to our gluconeogenesis. So sometimes our cells or certain parts in our body may be starved for energy and may not have enough sugars which they can utilize for the ATP production. Well, in this case, our cells actually have an internal control system so that they can go through a process, the process of glycolysis in reverse, and actually regenerate some sugar molecules in order to get our body back to homeostasis, at least for some time. And that process is called gluconeogenesis. It's basically, again, sugar new formation, right? In which our body cells can take those pyruvates that they've generated already. And before they can go back, go into the citric acid cycle and go further, they can kind of work through the process in reverse and get some of those sugar molecules back in the system. However, this process this is also sometimes needed when there is a high energy input or high demand for glycolipids or specific sugars for glycolipids or for glycoproteins needing to be made, or the cell is undergoing cell replication or DNA replication, which is requiring a lot of these glucose 6-phosphates to make those nucleotides. And so in that case as well, you will have some level of this equilibrium happening where some of these pyruvate molecules are actually converting back into these intermediate steps in order to get those intermediate molecules that are needed for those processes to occur. Usually this does require, again, an ATP input that is needed to make these processes continue to work. However, it can happen, and it does happen, both to keep our body at equilibrium with a certain level of blood glucose level, for example, and also to provide building blocks to our cells when needed for the processes that they need to have happening at that time. So some of these processes can happen just the way we talked about. They essentially work in reverse. However, some glycolytic steps cannot be reversed in gluconeogenesis. So an example of that is shown right here, which is the process with which you get the fructose 6-phosphate into a fructose 1,6-bisphosphate. So that second phosphate that is added onto your fructose. In glycolysis, it would have taken that ATP and it would have transferred that back into the phosphate, into an ADP as it took that phosphate off. Now, when this works in reverse, it cannot just take ATP and convert it back into ADP. Instead, it actually goes through a different release by breaking using high water molecules, just like it would in any other macromolecule breakdown. It does so through hydrolysis. It breaks this down with a water molecule to release the phosphate from this fructose 1,6-bisphosphate to convert it back into fructose 6-phosphate. So you're not getting an ATP as a result. It cannot do that. Instead, you're just releasing the energy like you would in a typical macromolecule breakdown. Now, this fructose 2,6-bisphosphate is an important regulator inside our system to dictate how much glycolysis versus how much gluconeogenesis is happening at any given time. These are controlled by regulators, other regulators in our body, signaling molecules in our body that are either activating or inactivating one of these systems. And those molecules are glucagon and epinephrine. So you should know that the signaling molecules that would activate this regulator, fructose 2,6-bisphosphate, as a fructose 2 and 6-bisphosphate as regulators inside our cells, are glucagon and epinephrine. Okay, so those are your two signaling molecules that can control how much glycolysis is happening versus how much gluconeogenesis is happening. This requires, again, a signaling cascade, which activates several proteins. The two proteins that it activates specifically is PFK2 and F2 bisphosphate activates phosphatase activity, which will then phosphorylate the fructose 6-phosphate into fructose 2,6-bisphosphate. So these are protein kinase that are activated that then further phosphorylate the fructose 6-phosphate to create those molecules. Now, if you get PFK1 as a result, which is a single phosphate added to this fructose 6-phosphate, it activates glycolysis so that you have more glycolysis happening, more glucose getting broken down. In addition, the fructose 2,6-bisphosphate can also produce inhibit this phosphatase activity, which leads to inhibition of gluconeogenesis. So you can get further activation of glycolysis without returning back to more glucose states. So the more activation of this pathway occurs, the more glucose is going to be broken down and the less gluconeogenesis will happen. Does that make sense? Right. So this the activation of this pathway leads to activation of glycolysis and more energy being produced by the cell.
And
so in this case, yes.
Is this happen all the time?
Yes. So there's always some level of activity for these happening at all times. So in an example where you have equal amount of gluconeogenesis and glycolysis happening, you would have similar amount of PFK1 and F1 phosphatase activity in your cell. In places where you have more glycolysis happening, you will have activation of PFK1 more at a higher level than the inhibition of the phosphatase activity. Okay.
Okay.
Yes, no. Yes, it's happening all the time, but the levels differ, right? So how much inhibition versus activation is happening in the system is going to be dependent upon how much glucagon or epinephrine is getting released onto your cells to bind to the receptors and activate that response. Yes, so when your body triggers the flight or fight response, it's basically saying, just break down all the glucose. That's why you have that release of energy as a response. Exactly. Good example, Ryan.
And what about
I'm going to use it next time myself.
What about if we are dieting, we're cutting up sugars?
So the issue with the dieting part is that then you have not enough sugars in your body to utilize. Your body actually goes into a very different system, metabolic pathway. So it is going to be breaking down some of those fatty acids. And so those are regulated in a different way. So when we go back, let me go back to that step. Actually, we're going to talk about that soon too. But when you look at this pathway for acetyl coenzyme, right there. So what you're getting in those cases are those fats getting released from the fat droplets as part of the system. And those, because you don't have enough sugars, it's going to start using some of these fats that are stored in our system and breaking them down through citric acid cycle. But it will also use a lot of those amino acids that are, or the proteins that are coming in, and use them to create pyruvates and activate that system. So they have a very different activation response than our typical glycolysis itself. So this specific process is specifically for glucose breakdown and control of how much glucose is getting released and how much glucose is getting used.
Because I have read one time that if you are dieting on high protein, so your body is making sugar, maybe the pyruvate is
You do
activated.
create, well, yes, that's the thing, that no matter what you are doing, right, how you're consuming your food, you can have a high protein diet, you can have a high fat diet, you can have whatever mixture of diet. At the end of the day, remember that your body still has to convert it into one of those steps that can fuel the citric acid cycle so that it can fuel the electron transport chain. So if it cannot get glucose as glucose, it's going to break down those other sugars into glucose, or it's going to break down the amino acid or fatty acids into either acetyl-CoA or a pyruvate so that it can fuel that same reaction. Right? So you can get energy out of proteins by just breaking them down into amino acids. You still have to convert them into one of those molecules in that chain to get to the citric acid cycle or to get to the electron transport chain.
Are we like, when we do this, are you like working out our cells more? It's bad, or it's like okay to happen?
It's different, and there are going to be two sides to the coin. So some of these processes release a lot of oxidative damage in our cells, reactive oxygen species that aren't good for our body, and that can lead to aging response beyond what you typically see. But doing it for a short period of time, anything, you can do that and it would not be harmful for your cells.
Okay, thank you.
Okay, so now let's talk about how do we utilize those storage molecules that we have in our body as energy. So animal cells obviously use glucose stored in the stored form. It is in glycogen, which is essentially these large polymers, right? It's a polymer or polysaccharide chain that is made up of several of these glucose molecules joined together, making what we call a glycogen polymer. And these are going to be stored in granules in our liver cells and the cytoplasm of our liver cells, as well as in some other parts of our body as well. Now, this glycogen gets broken down. This long chain of glycogen can get broken down into individual monomers through addition of phosphate groups to those to create, again, high energy bonds. And the ones that it will break them down into is usually a glucose 1-phosphate or a glucose 6-phosphate, which essentially indicates the places where it is making those bonds to begin with. So as it is broken down, it is broken down by a actual enzyme, glycogen phosphorylase, which phosphorylates these bonds and breaks as it breaks them down. Now, these are then thrown into the glycolysis process because both of these are essentially intermediary steps within the glycolysis process, and they can just be used directly into it to go through the same cycle as we've talked about before, the same catabolic reactions. And so this is then continuously repeated as long as you need that extra glucose available from the glycogens. The other way that our body stores energy is in the form of fats that are stored in these droplets inside the fat cells. Now, the fats are stored inside the cells in many different ways, right? And they are required by many different parts of our cells as well. So you have your phospholipid bilayer, and many of these fat droplets are going to be right inside that space, again, in the cytoplasmic space, as either triacylglycerols or as cholesterol esters that can then be utilized by our cells. Plants, on the other end, are going to have both starch and fats in their chloroplasts. That's where they store their energy. And again, oil droplets are going to be the way they store their fats, and starch is going to be their energy storage system inside the chloroplast in the cell. So it's not in the cytoplasm, but rather in the chloroplast within that cytoplasmic space. And that way it is very readily available for energy consumption for these plants. Now, in addition to that, seeds of plants often obviously contain large amounts of fats and starch that can not only be used by the plant themselves for energy consumption, but also can be used by us as consumers or of this energy as well. So the way our metabolism is regulated is through coupled reactions, where catabolic and anabolic reactions are organized and regulated in a chain format. And you can have feedback regulation to allow cells to go from breakdown of these macromolecules to release energy, or to make those macromolecules to store energy, depending on the need of the cell and what kind of functions it needs to perform. And cells store these molecules in special reservoirs as needed until there is shortage of those molecules, and then they can use them for energy consumption. Questions? Oh, we did good. We got through this chapter actually within the time frame.